Hello, and thank you for joining us today for the latest The Scientist webinar. My name is Iris, and I'll be moderating today's event. I'm very pleased to introduce our speaker, Crystal Emery. Crystal Emery is the product manager for Lunatic at Unchained Labs. She holds a Bachelor of Science from UC Santa Barbara and a Master's from San Francisco State University. In previous roles, she worked hands-on with next-generation sequencing workflows and supported semi-automated laboratory technologies and applications. She enjoys collaborating with researchers to streamline complex workflows and innovate instrumentation that drives efficiency in the lab. A very warm welcome to you, Crystal. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system to the side of the live stream player. And with that, I'll now hand over to our speaker to begin their talk.
Thank you for the introduction, and hello to everyone joining us today. I'm looking forward to sharing how you can make NGS your BFF with Lunatic. NGS library prep workflows often have a lot of steps and a lot of opportunity for things to go sideways. And ultimately, if you get to the end of your library prep and sequencing fails, you have to begin the tedious process of troubleshooting and maybe even starting over from the beginning. Just like making the perfect friendship bracelet with finicky little beads and string that doesn't sit quite right, realizing that you made a mistake and have to start all over is pretty devastating. That's why it's so important in a complex workflow to make your steps as simple as possible and to be able to check your progress to confirm everything looks right, starting from the very beginning. There are a lot of ways that NGS library prep workflows are done, depending on the goal. Some workflows require PCR, some don't. Some require extra capture and cleanup steps, some require extra size selection steps, but they pretty much all start at the same place, with extracted DNA or RNA of unknown concentration and quality. Many labs use fluorescence-based assays for determining nucleic acid concentration at the beginning of their library prep workflows, but these assays have a lot of steps. They require prepping dilutions and standard curves, use costly light-sensitive reagents that degrade over time, and introduce the risk of pipetting errors. And since dyes are only measuring the nucleic acid content, you're missing out on the detection of potential contaminants that can inhibit your downstream assays. Considering how many samples you're multiplexing, it's worth asking if there's a faster, simpler way to get dependable quant and QC results. And if those samples you're multiplexing happen to vary in concentration, fluorescent dyes add another wrinkle. They're only linear across a narrow range. Push past it, and they saturate, complicating the workflow by forcing you to dilute your samples. Without knowing the starting concentration, though, you're guessing at how much to dilute. More guesswork, more steps, more chances for inaccuracies that can quickly turn into poor sequencing results. With Lunatic, you know immediately that your samples are in range. The data here shows Lunatic maintaining linearity across a wide concentration range. In the lower picogreen compatible range, both assays agree closely until picogreen saturates. That limited dynamic range can mean diluting a sample multiple times just to land on the standard curve. Lunatic sidesteps all of that, delivering reproducibility and accuracy across a wide dynamic range, no standard curves or serial dilutions required. Lunatic is a full-spectrum UV-Vis spectrophotometer which requires only 2 microliters per sample and no dilution steps, reagents, or standard curves. Lunatic can measure up to 96 samples at a time in about 5 minutes. Our plates have a standard SBS footprint and a standard well layout, so they're compatible with liquid handling robots and multi-channel pipetters. Unlike fluorescent dye quant, Lunatic gets results in a single step. Just read the samples and move on to your library prep's downstream steps with your accurate quantifications in hand. The Lunatic plate is what makes this amazing capability possible. It has the same well spacing as a 96 well plate, but instead of just a well, each one is connected to a microfluidic circuit. Each microfluidic circuit has either 1 or 2 fixed path length microcuvettes, which cover a wide dynamic range of 0.03 to 275 OD. These circuits ensure that there's no cross-contamination or evaporation. Fixed path lengths give a high level of precision and accuracy to absorbance measurements, which means you know exactly what you're measuring. While we just need to take a single step with Lunatic, just load and go, a few more things happen behind the scenes. After your sample lands in the well, it's pulled into the reservoir by capillary action. At this point, the sample can sit for up to 2 hours before reading, with no evaporation. That gives your workflow a bit more flexibility, especially if you're running lots of samples in a day. Then, you load the plate into the instrument, which reads the empty microcuvettes to eliminate any absorbance contribution from the plastic. Then, a pump applies a small vacuum, which moves the sample into the microcuvettes for reading. Here we can see exactly what sample loading looks like with the help of some fluorescent and a black light. In 2 wells, we've already added 2 microliters of fluorescent, and as you can see, to load another sample, you simply add in 2 microliters to that input well, and the sample wicks into the plate. Here's what it looks like before and after a read happens. You can see the fluorescent in the microfluidic reservoirs on the left, and on the right, the samples pulled into the microcuvettes. You might notice that one cuvette is brighter than the other. That's because one of them has a longer path length than the other, which means there's slightly more volume of the sample in that one, so it's more visible. This combination of 2 different path lengths is part of what gets Lunatic its phenomenal dynamic range. Lunatic easily meets the needs of labs working in larger scales, like clinical diagnostics labs and core facilities, for example. The plates used for Lunatic are SBS format and are compatible with liquid handling robots and plate grippers for fully automated workflows. In some labs, Lunatics are integrated in work cells for extraction, quant, and QC, for various library prep steps, and for automated normalization and pooling before sequencing, seamlessly processing thousands of samples per day. I'm sure you're wondering, how well does a mere 2 microliters really load into these plates? As you can see, easily. The plates are in standard SBS format, so they work with whatever liquid handling platform you prefer. If you're going for a full integration, Lunatic also has an API software option that lets you take control. The plates can also be tracked with barcodes, ensuring traceability in certified labs. Lunatic brings in 2% accuracy and 1% precision for its absorbance measurements, which we've validated in several ways using independently verified standards. First, we have these 3 NIST reference materials we can use to verify Lunatic's absorbance results: a monoclonal antibody, a tryptophan standard, and a uracil standard. In every case, Lunatic measurements of the standards were within 2% of the NIST certified value, and CVs within 1%. You can read about it in this app note on our website, Spot on UV-Vis Accuracy with Lunatic and Stunner. NIST also has SRM 2372A, a human DNA quantitation standard mainly for qPCR. It consists of 3 components, each of which has a certified mass concentration and a 95% confidence interval. As you can see, Lunatic's measured concentrations were within the confidence interval and had CVs of 1% or less. Component A is from a single male donor, B is from a single female donor, and C is a 3-to-1 gravimetric mixture of male and female donor. When you look at Lunatic's linearity, accuracy, and precision across a wide range of concentrations, the performance really stands out. This DNA dilution series spans from 25 to 1400 nanograms per microliter, and you can see how closely the measured values track with the expected concentrations, showing off Lunatic's spot-on accuracy within 2%. At the same time, the low CVs across the entire range, typically below 1%, highlight Lunatic's exceptional precision. Delivering this level of dynamic range, linearity, accuracy and precision is the advantage of using fixed path lengths. Lunatic handles high concentration samples without dilution, with high accuracy and reproducibility. The UV spectra shown here have the telltale absorbance peak at 260 nanometers, which is indicative of purified nucleic acids. In this example, the DNA dilution series on the left reaches up to 12,800 nanograms per microliter, while the RNA series on the right goes up to 6,800 nanograms per microliter. These concentrations are higher than what's typical for genomic samples, but definitely in the range for synthetic DNA, like your indexing oligos. To support this range, Lunatic offers two plate options. High Lunatic plates extend the dynamic range from 0.03 to 275 OD, making them ideal for extremely concentrated samples. Standard Lunatic plates reach up to 40 OD, which corresponds to about 2,000 nanograms per microliter of double-stranded DNA. Both plate types run on the same instrument with no additional setup or software changes required. Since DNA and RNA extractions can be super messy, I want to show you how Lunatic's unmix applications let you see annoying impurities that other UV-Vis systems would mistake as additional DNA or RNA, and that dye-based quant would just miss entirely. Here, we're looking at results from a sample of purified DNA in TE buffer that are pretty close to ideal. This is a typical result for a clean sample. We can see the expected peak maximum at 260 nanometers, and quality ratios are within the acceptable range. For pure DNA, we would expect an A260-A230 ratio greater than 2, and an A260-A280 of about 1.8. Note the accuracy and reproducibility. We're seeing nice low standard deviation and percent CVs for triplicate reads. For a second example, here's DNA in TE buffer with excess EDTA, about 5.5 millimolar. At this level, EDTA can inhibit polymerases needed for PCR and library prep, ultimately leading to sequencing failure if not addressed. The good news is that this can usually be fixed by repurifying the sample and re-eluting. And in this case, the DNA yield is high enough that repurification makes sense. But if the yield were low, it might be better to discard the sample altogether. Unmix helps you make that call by deconvoluting overlapping absorbance signals, revealing both the true DNA concentration and the source of the abnormal signal, so you can troubleshoot effectively and make informed decisions about your valuable samples. With Lunatic's intuitive and simple software interface, it's easy to get a quick glance at your results and know exactly what's happening with your samples. You can view a full plate of samples at once, or dive into the details of each sample to get a closer look. In this detailed example, we can see the measured absorbance spectrum in black, the contribution from impurities in blue, and the DNA peak in purple, reflecting the true nucleic acid concentration. All of your data can be exported in a single click, whether as a CSV, Excel file, etc., ready to drop directly into your preferred LIMS. And in fully automated environments, results can be uploaded and integrated into your workflow immediately, enabling real-time decision-making and rapid calculations for downstream workflow steps. This easily exported data is especially handy at that stage in the library prep process, commonly referred to as normalization. When you have hundreds of barcoded samples to pool into one high-throughput sequencing run, they must be combined at equivalent concentrations. Add too much of one sample and it'll steal sequencing reads from the rest of the multiplex samples. Add too little of a sample and you won't get the sequencing depth you need. Getting normalization right is absolutely critical to ensuring you don't have to repeat sequencing runs. Most protocols will suggest you use dye-based methods for quant before normalization, but dye-based quant isn't always as accurate as you might think. Here, you can see the outputs from one of our biobank customers, where they compared the results of samples normalized with Lunatic and picogreen. They quantified thousands of genomic DNA samples with either method, normalized them to 25 nanograms per microliter, and measured again. You can see there's a much wider variation in the results with picogreen compared to samples read on Lunatic. When you have to dilute your sample to fit your standard curve, you're going to see more error and more risk of poor normalization, which ultimately means more risk of sequencing failures. On the other hand, Lunatic quickly and easily measures concentrations for your normalization calculations without wasting any plate space for standard curves or worrying about dye saturation and dilution inaccuracies when your concentrations vary. Lunatic's stellar precision and ability to read a wide dynamic range of concentrations makes it consistently reliable for building balanced pooled libraries. Just read the ODs of each sample, perform the normalization calculations, and pool accordingly. And if you want to reduce risk of errors even further, take the human out of the equation entirely. Here, we're showing how the Unchained Labs automation platform, Stuntman, can use an integrated Lunatic to measure concentrations for a full plate of multiplexed samples and automate the normalization and pooling in preparation for sequencing. We start by telling Stuntman what to do in plain language and let it take charge of the workflow design that will happen on deck. With automation being driven in a Python environment, changes are flexible, deterministic, and traceable. Stuntman begins the protocol by pipetting samples into the Lunatic plate, loading the plate onto the Lunatic instrument, and measuring the UV-Vis absorbance. To get an equimolar pool of the multiplexed samples, we can simply normalize by OD as a standardized way to bring the samples to the same concentrations. Whether you prefer to dilute your samples to equivalent concentrations and then pool, as shown here, or pool by aspirating and combining unique volumes from each sample, Stuntman can do it. All automated end-to-end from concept to design to execution to analysis. Researchers around the world are using Lunatic in their NGS workflows. To give you one interesting example, the UK Biobank, an open resource with genomic and phenotypic data on approximately 500,000 individuals, used Lunatic as part of their process to check both concentration and purity of extracted DNA. Their process involved collecting blood, isolating the buffy coat, and extracting DNA using magnetic bead extraction. That DNA was then split into 3 aliquots, 2 for storage and 1 reserved for genotyping. The primary aliquot was quantified on the Lunatic as a quality stand-in for its sibling aliquot. If it met their criteria, DNA concentration above 10 nanograms per microliter and an A to 60, A to 80 ratio above 1.8, the sibling aliquot proceeded to genotyping. The genotyping results had an average call rate of 99.48% and a pass rate of 99.45%, confirming high-quality DNA across the cohort. This UK Biobank study also compared Lunatic to picogreen across nearly 500,000 samples, and the results showed a strong correlation in DNA quant between the two methods, validating the accuracy of this UV-Vis approach at massive scale. Most importantly, they found no difference in the quality of the downstream data depending on the measurement technique. And unlike picogreen, Lunatic also provides purity information in addition to concentration, and costs about half as much per sample. These findings were published in Welsh et al. In 2017 if you'd like to dig into the data for yourself. Another example is the All of Us research program, sponsored by the NIH, which works with the Mayo Clinic Biobank and aims to enroll 1 million or more participants into a resource combining health records, biospecimens, and genetic data. In their workflow, DNA was extracted from blood using either salt-based precipitation or a magnetic bead-based method. Then, DNA concentrations, along with A260, A280, and A260, A230 purity ratios, were measured on the Lunatic. Samples meeting a minimum concentration of 50 nanograms per microliter and A260, A280 purity ratios of 1.6 to 2 moved forward to normalization, library prep, and whole genome sequencing. This ongoing project required an investigational device exemption from the FDA, and the team published a 2022 paper detailing that validation process, including how the Lunatic was used for sample QC. The key takeaway here is Lunatic is the ultimate NGS quant tool. It's simpler, faster, and easier than other methods, without the need for dyes, reagents, or dilutions. It helps QC your samples in addition to just quant, and it's fully automatable for your highest throughput needs. With Lunatic there to save your workflow from tedious steps and downstream problems, you might even start to think of NGS as your BFF. Thank you for your time. I'll leave it there for now, and I'd be happy to take any questions.
Thank you very much, Crystal, for that wonderful presentation. We really appreciate you sharing your expertise with us today. I'm now going to move into the Q&A session. To submit a question for our speaker, please enter your question into the box to the side of the live stream player and click send. We'll answer as many questions as possible during the time available. We have already some questions submitted, so I'll move right into those. Crystal, the first question is, what is the concentration range for DNA and RNA on Lunatic?
Yeah, so the concentration range for DNA using a regular Lunatic plate would be 1.5 nanograms per microliter up to 1,600 nanograms per microliter. Sorry, 2,000 nanograms per microliter. RNA goes up to 1,600 nanograms per microliter from 1.3 nanograms per microliter. On the high end, with our high Lunatic plates, DNA can go up to almost 14,000 nanograms per microliter. And the RNA goes up to 11,000 nanograms per microliter.
Thank you. And is the plate single use?
The wells are single use, but you can run up to 96 different experiments if you want to do just single use at a time. So you can run the plate as many times as you need to use up all of the wells on the plate.
Thank you. And our next question is, can Lunatic quantify protein as well?
Yes, it absolutely can. It would use the A280 absorbance wavelength to measure protein. And we have a variety of applications as well for protein use cases, including unmixed applications to pull out different impurities that are more common in protein workflows.
And is there a need to run blanks with Lunatic?
It's always good to run a blank, yes. We also have a way to store blanks. So if you're running the same samples with the same sort of buffers repeatedly, you can store those blanks in the system and we can reference that spectrum. For the unmix strategy of applications, we would recommend using a water blank. And for the standard applications, you would just use a buffer blank as usual.
Thank you. And our next question is, which automation platforms would Lunatic be compatible with?
We've partnered with a lot of different automation platforms before, so I can pretty confidently say pretty much whatever you're using, the Lunatic can work with it, whether it's Hamilton, TCAN, OpenTrons, Revity. Sorry if I've missed you. We've pretty much done it all. Obviously, Stuntman with our new automation platform through Unchained Labs is our preferred favorite, but because it's in that SBS plate format, the Lunatic plate can be loaded or moved with any platform you choose.
Thank you. And what contaminants can Unmix detect?
It depends application by application, because it's going to look for different impurities based on that application. And if you want more detail on that, we've got a great application note on our website. It's called A Look Under the Hood of Lunatic.
Thank you again, Crystal, for joining us today. Unfortunately, that's all the time we have, but any questions we didn't manage to get to will be answered offline as soon as possible. The webinar will now be made available on demand, and you can access the recording using the same link you used today. We'll also make sure any questions submitted on demand are passed along. A certificate of attendance will also be sent by email to everyone who attended today's session. That just leaves me again to thank our speaker, Crystal Emery, and to everyone in the audience. See you again soon.